I am an ADC. My potency is affected by a key metric, the DAR value, the number of payloads I carry. Inside the body, my DAR can decrease due to cleavage caused by metabolism or chemical instability. Monitoring this shift is vital for efficacy and safety. Traditional methods like ELISA can detect me, but they cannot evaluate the payload release accurately. Even the free drug analysis may not reflect true payload release. In the complex in vivo environment, conventional analytical methods fall short. A new strategy is needed. WuXi AppTec DMPK's qualitative and quantitative dual strategy solution is designed to precisely map my dynamic DAR in vitro or in vivo. The first step is precise immunocapture, isolating me from interference. Strategy 1. Full profile. HRMS scans my intact form, clearly showing the mass distribution and relative proportions of antibody fragments. Profiles over time indicate my metabolic fate, linker cleavage, or payload loss. Strategy 2. Precise disassembly. Specific enzymatic cleavage breaks me into antibody fragments and payloads. LC-MS then provides ultra-sensitive quantification. A simple ratio yields my precise average DAR. This method is sensitive, fast, and robust. Qualitative methodology shows where I have changed and what I have become. Quantitative methodology determines how much I have changed. Together, they reconstruct my in vivo DAR evolution, providing a solid data foundation. As I progress from discovery to clinic, DAR is one of the critical quality indicators. WuXi AppTec DMPK brings deep expertise in conjugated therapeutics from hundreds of projects. We deliver precise mapping to guide your drug to success.